Let's talk about the cell membrane. And the cell membrane serves several important roles, and it provides the structure of the cell, and also it controls the material transport across the membrane. So let's first look at the structure. It's called a lipid bilayer. Lipid bilayer means you have those lipids which forms the cell membrane. So each lipid you have head and a tail. The circular thing I draw is a head, and the two line uh, are the tails. So the head is hydrophilic, means they are happy with going with the water, and the tails are hydrophobic, means they don't like to be in contact with water. And for this reason, we have bilayer, means two layers of lipids. Because the tail, they are hydrophobic, they cannot be directly in contact with water, and that's why they want to stay inside, like being away from water, etc. So here, this is lipid bilayer, also called plasma membrane. So outside, we call it extracellular space. And on the other side of the membrane is the plasma. Or we call it cytoplasm. Cytoplasm. Maybe this is a better word to call it. So cytoplasm is what inside the cell, and extracellular space is what outside the cell. So this lipid bilayer forms like a two-dimensional fluid, and they also allowing lateral movement of the lipids and the membrane proteins to sort of diffuse with it a little bit. So this is the lipid bilayer. So what I'm drawing is, let me put it into context. This is a cell. And what I'm drawing is a part of the membrane. So this is what I'm drawing. And on the membrane, it's not just a lipid bilayer. They have a lot of proteins. So let me draw some proteins on the membrane. Proteins are usually embedded in the membrane, and they come with different shapes and different functions. Just drawing 3 to illustrate that proteins comes with different configurations, morphologies, and functions. So some of the proteins, they serve the purpose of like signaling, and some of the proteins function as provide a structure, or some like as a gating mechanism. So the lipid bilayer itself are not very much permeable to substance. And if some substance like electrolytes or proteins, or not necessarily protein that doesn't transport in a different way. It's most likely like those like molecules, like glucose, those type of things. If they want to go across the cell membrane, they depending on what they are, they may need the help of a protein to help them transport. So we will get to that. And then now let's talk about membrane permeability. When we talk about membrane permeability, we need to recognize that they have hydrophobic tail. So um, phobic core. And because of this, it acts as a barrier to most polar and water soluble molecules. The hydrophobic core form by a barrier most polar and water soluble molecules. So a lot of biologically relevant molecules are indeed polar and water soluble. That's why they need some help. But for small molecules, uncharged small molecules such as like polar, give a small list. Polar hydrophobic molecule is such as they need to be small, such as oxygen, nitrogen. They can dissolve and is permeable to the membrane. But it's not, the membrane is permeable. Let me think on it. Can dissolve in the membrane and can be transported across the membrane without proteins. Be transported across the membrane without proteins. Non-permeable like when you put like charged ions, maybe charged ions, they are they cannot go through the membrane. So membrane are not permeable to it. So they are not permeable to the membrane. charged ions such as calcium chloride, calcium bicarbonate, etc. need specific for transportation. Okay, now let's look at another concept called membrane potential. We will go back to the membrane proteins like ion channels, pumps, and the co-transporters in our next lecture. But right now, let's just go through the overall structure and some basic properties of the membrane. So let's also talk about a concept called the membrane potential. We will need it in the next lecture. Membrane potential. Because the cells inside and outside, there's a lot of like electrolytes. And collectively, they contribute to the voltage difference across the cell membrane. And it's not just about the electrolytes, it's also mostly it's because of the lipid bilayer. So we just draw the lipid bilayer. Those lipids, they are not necessarily like electroneutral. For some of them, about like 10% of them or something, like they are charged. And especially you have two layers, the in layer, the layer that is facing the cytoplasm, some of the pads and electron also of the lipids, they are negatively charged. And then those collectively contributes to the membrane potential. So the membrane potential is let's define this is the voltage difference across the cellular membrane. So this is across the cell membrane. So we usually give it a notation called Vm. M refer to membrane. So we define it as, it is a convention. We define it as the voltage in means the voltage inside of the cell minus the voltage outside of the cell. So that's we call the membrane potential. This is the voltage difference across the cell membrane. And also by convention, we set V out to be 0. And in that case, Vm membrane potential is just the the voltage inside of the cell. So Vm equals to V in. So you usually when people refer to membrane potential, give a value, people usually also understand it as the voltage inside of the cell because outside is set to be 0. So we set V out to be 0 as a reference point. So for most tissue cells under physiological conditions, for most tissue cells under biological conditions. Vn is about minus 16 millivolts. Again, this is under some like normal conditions. And in a lot of cases, for example, for cancer cells, or for cells under pathological conditions, or for during neuron failing, and in a lot of cases, the membrane potential can be different from this value, and sometimes they may be depolarized. That means they become less negative, for example, become minus 30 millivolts, for example, to be a depolarization. And then this is, let me put down. If Vn changes from minus 60 millivolts to minus 30 millivolts under some environmental perturbation. This is a process called depolarization. So when cells has negative voltage inside, it's considered as polarized. When it becomes less negative, it means depolarized. That is the language used in this field. So before we stop this lecture, let me just mention the unit of or we put the membrane potential, because we will need it in the next lecture when we are going to do some calculation. So from physics, you know that voltage has a unit called volt. It's called V. And well, volt. So 1 volt. So volt itself is not a fundamental dimension. It's actually a derived. That means it is interpreted from other more fundamental or other combinations of units or dimensions. So 1 volt is defined as 1 joule coulomb. Joule is the unit for energy, and coulomb is the unit for charge. So it's the same as 1 joule per coulomb. So this is like how you interpret the physical meaning of voltage. So voltage is like an energy, but it's an energy with respect to how much charge you have. So it's energy a coulomb. So this is the interpretation of voltage. All right, that's all I would like to cover for the cell membrane. Cell membrane is way more complicated than what I cover here, but what I covered is what we are going to use for the rest of this class. So what is needed for this course. And feel free to read more about cell membrane if you are interested.